In this video, I will be discussing sex determination in fruit flies, otherwise known as Drosophila melanogaster. There are a variety of different male and female specific morphological differences, including but not limited to the overall size, shape, and color of the abdominal tip. The sex of Drosophila is determined by the X chromosome to autosome ratio, and the sex-specific genes that are controlled by the X chromosome to autosome ratios are subjected to alternative splicing. The central dogma outlines the sequential flow of genetic information from DNA to RNA to protein through transcription and translation. Eukaryotes separate transcription and translation both spatially and temporally, with transcription occurring in the nucleus and translation occurring in the cytoplasm. Pre-mRNA undergoes processing, including the addition of the 5' cap, splicing of introns, 3' cleavage, and the addition of a 3' polyadenylated tail to become mature mRNA. These RNA processing events regulate mRNA export, stability, localization, and translatability. Alternative splicing is a crucial mechanism in eukaryotes, as it enhances the genomic coding capacity and enables the production of multiple mRNA transcript isoforms from a single gene. Alternative splicing is a key mechanism that controls sex determination in fruit flies. Sex-specific genes in Drosophila rely on X chromosome to autosome ratios, 0.5 for males and 1.0 for females. There are three major downstream target genes in the sex determination pathway: a sex-lethal splicing repressor, SXL, a transformer splicing activator, TRA, a double-sex transcription repressor, DSX, which produce proteins crucial for regulating sex-specific phenotypes through alternative splicing. Males Drosophila adhere to the standard sex determination pathway, and so we will address them first, starting with the SXL mRNA transcript. In males, a third exon containing a premature termination codon is included in the SXL transcript due to the activity of the splicing factor SPF45, leading to the non-functional SXL protein being made. In the absence of functional SXL protein, TRA transcripts undergo male-specific splicing that leads to a longer version of exon 2 with the premature stop codon being included. Splicing factor U2AF binds to the 3' end of the TRA intron in males, encouraging splicing at the proximal site of the second exon and leading to the non-functional TRA protein being made. The lack of functional TRA protein results in male-specific splicing of the DSX pre-mRNA. In the DSX mRNA transcript, male Drosophila have a weak 3' splice site between the third and fourth exons. So the 3' splice site is not recognized and exon 4 is spliced out. This results in exon 3 and 5 of the DSX transcript being spliced together. The DSX mRNA transcript isoform that forms because of this splicing will act as a transcription factor that inhibits the expression of female-specific genes in other areas of the genome. In females, a functional SXL protein autoregulates its own splicing by binding to the mRNA transcript into splicing factor SPF45, inhibiting SPF45 and resulting in the exclusion of the third exon and production of more functional SXL protein. The functional SXL protein will then bind to the 3' splice site of intron 1 in the TRA pre-mRNA, forcing splicing factor U2AF to promote splicing at an alternative downstream splice site and leads to the production of a functional TRA protein. This functional TRA protein will then recruit proteins TRA2 and RNA binding protein RBP1 to the exonic splicing enhancers on the DSX mRNA transcript and regulate its splicing. TRA, TRA2, and RBP1 will then induce splicing at the 3' splice site of the DSX mRNA transcript and facilitate the inclusion of exon 4. As a result of the splicing, a female-specific spliced isoform of the DSX protein is produced and will go on to act as a transcription factor to suppress the expression of male-specific genes throughout the Drosophila genome. Thank you for watching, and goodbye.